Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. We will speak now about the viral diseases of camels, management of diseases outbreaks in livestock farms. Viral diseases of camels. We will discuss today Camel Contagious Ecthyma, CCE. Camel Contagious Ecthyma has another name, Camel Contagious Pustular Dermatitis, CPD. Camel Contagious Ecthyma is a highly contagious viral disease, mostly of young camels, that contributes to calf debility and occasionally calf losses. Camel calves born during the same calving season could develop CCE. The pathogenic risk factor. Several studies showed that CCE viruses are closely related to pseudo-cowpox virus species of the parapox virus genus of family Poxviridae. The virus virions are ovoid particles that range in size from 230 to 360 in long and 131 to 160 in width nanometers. Their axis ratio is about 1 to 1.56 and their surface shows a regular cross pattern of filaments, pole of wool, under the electron microscope. There is antigenic diversity between CCE virus and other related parapox viruses like orthovirus and bovine papular stomatitis virus, and camels were not protected after immunization with a vaccine against sheep and goat contagious eczema. CCE virus withstands to 56 centigrade for an hour, pH 3, from 8 months up to a year at 25 centigrade in dried escapes. CCE virus is susceptible to various disinfectants such as 1% carbolic acid, 1 to 2000 formalin, 1 to 20,000 mercury chloride. Susceptibility. The age group at risk are those less than 1 year of age, including cases reported in a month-old camel calves. CCE is not only a cause of calf mortality, but also affects camel performance with weight loss and severe reduction in milk production, since she camels mostly ceases to lactate when their calves die. Mortality rate. In most cases, CCE caused no mortality, but when camel calves are severely affected, the ecthyma lesions interferes with calf ability to suckle or graze and extends to eyelids, leading to blindness, particularly at the savanna pellet in Africa, leading to mortality rates that can reach to 9%. This disease usually passes off without causing death, but at 6.6% herd mortality and 3.8% case fatality rates have been reported. Geographical distribution. Camel herders in many countries regarded CCE disease as a separate disease and gave it local names such as Osdok in Kazakhstan, Ambror in Ethiopia, Abu Shalambo and Al Kulayt in Sudan. Contagious pustular dermatitis in camels was first described in Kazakhstan in 1968 and has since been described in many camel rearing countries as in the following map. In this map, we see countries reported outbreaks of camel contagious ecthyma. Take the red color. Morocco, Nigeria, Libya, Sudan, Ethiopia, Kenya, and Somalia in Africa. In Asia, Saudi Arabia, all the Gulf countries, Iran, Iraq, Turkey, east of Europe, Russia, India, and Bangladesh. Disease transmission. In the following figure, we will discuss the geographical distribution of camel pox disease all over the world. This diagram illustrates the transmission cycle of camel contagious ecthyma. These browsing on trees with spikes, the virus in dried crusts survive longer in the environment, so these trees cause wounds in the face and lips due to spikes, so the virus is transmitted to camels. Then, injury during tagging, ear tags, or lesion in lips would be seen. Then from camel to camel through aerosol transmission. And also oral gavage tube or injury during gavage will transmit the disease to parasites. Also, wild animals infected with ORV is transmitted to domesticated animals, including camels. These viruses will transmitted to human through owners or milkers or veterinarians that did not kosherly treated the infected animals. Transmission of camel contagious ecthyma occurs through direct and indirect contacts from contaminated environment and between sick and susceptible animals. Watering camels at water boreholes and dug out water reservoirs provides opportunity for infection transmission as contagious camel ecthyma shed from affected camels contaminate water that becomes source of infection. Skin abrasions of the lips caused by browsing thorny trees seems to represent the major predisposing factor to camel contagious ecthyma. The thorny plants damage the lips, allowing virus penetration and multiplication. Movements of camels during the rainy season have a significant role in spreading of camel contagious ecthyma, and insects have also been incriminated in the spread of this disease. The marked seasonality associated with the rainy season may be due to the optimum condition for the survival and premutation of the virus and the skin abrasions caused by browsing thorny trees. Pathogenesis of Camel Contagious Ecthyma. Infection with Camel Contagious Ecthyma is confined to the skin epitheliotropic. The virus rapidly replicates in the skin, including hair follicles and sebaceous glands. These lesions mature into crusty scabs that eventually dry up and fall off around 4 to 7 weeks post-infection, but sometimes the course of the disease extended up to 3 months. The skin surface is largely indistinguishable from that pre-infection. When the scabs are peeled off, they leave an elevated raw bleeding surface that is prone to secondary bacterial infection, resulting in local tissue swelling, erythema, pain, and foul smelling. Clinical picture of Camel Contagious Ecthyma. Incubation period reaches to a week. It is primarily affects young animals exhibiting most regularly localized lesions and frequently generalized changes that resemble camel pox. It was formerly thought to be a form of camel pox since clinical symptoms of both diseases are similar, so it is very difficult to differentiate between the both clinical. Camel Contagious Ecthyma disease is characterized by the sudden onset and rapid progression of the lesion and proliferative crusty scaby epidermal lesions around the mouth and nostrils, usually accompanied with slight fever. Clinically, pox-like lesions first appeared on the lips of affected animals as small papules, followed in most cases with swelling on the face and sometimes extended to the neck. Papules progressively develop through vesicles and ulcers into scabs on the lips, muzzle, nostrils, and eyelids, culminating into fissured crusts on the lips. The lesion is proliferative and highly vascularized and may extend into gum, palate, and tongue. The pustules developed into fissured crusts that affect lips severely, leading to complete cessation of feeding or suckling, and this may reduce the body condition of the animal and its natural defense and increase susceptibility to other infectious diseases. Enlargement of the superficial lymph nodes, particularly the cervical and the mandibular, has also been reported in field cases. Affected animals lose appetite, are emaciated, and slightly anemic. At later stages of the disease, scabs become dark brown in color and drop off after 6 to 10 weeks. Figure number 1. An infected dromedary camel showed scaby lesions around lips, nostrils, and eyes. Figure number 2 of Camel Contagious Ecthyma showed an infected dromedary camel with 1.5 years old showed scaby lesions around lips. Another pictures from the Camel Contagious Ecthyma showed an infected dromedary camel with escapee lesions in the upper and lower leg. Figure number 4 shown an infected dromedary camel with escapee lesions on upper and lower lips with cutaneous fissures. A deceased camel with Camel Contagious Ecthyma that showed escapee lesions in the upper, lower lips and muzzle. A deceased bacterian camel showed hemorrhagic lesions and fissured crusts on upper and lower lips. The blue color here is due to treatment with gentiana. This figure showed a deceased dromedary camel with pustular and ulcerative lesions on the lips and eyelids with a pendulous lower lip appearance due to accumulation of the lesions. Figure number 8 shown deceased dromedary camels showed pustules and nodules as well as scars on the face, head, neck and shoulder regions. A diseased dromedary camels developed erythema and papules on lips with muzzle, and frequent lacrimation has been seen due to pain effect of these lesions. Figure number 10 showed dromedary camels developed swelling on lips, muzzle, and jaw, as well as intermandibular spaces. Figure number 11 showed dromedary camel with severe pustular lesions on the face and neck regions. This infected dromedary camel showed escapee lesions around lips, nostrils, and eyes. This collective figure showed representative clinical signs of camel contagious ecthyma in camels, where in figure A, proliferative escapee lesions on the lips and nose of a camel in a camel farm. Figure number B showed camel contagious eczema lesions around oral commissures in the camel. In picture C and D, severe bloody and multiple escapes lesion of camel contagious eczema had been shown. Laboratory diagnosis of camel contagious ecthyma disease. Sampling collection. Number 1. Tissue samples from lesions should be collected with a scalpel plate and placed in a suitable container in test tube or petri dish. The samples should be collected within 10 to 14 days from the appearance of the lesions. Samples can be maintained at room temperature. A 10 to 20% suspension should be prepared with phosphate buffer saline (PBS) with pH 7.2 and centrifuged at 2000 revolutions per minute for 10 minutes. These supernatant should be stained with phosphor-tungstic acid and then transferred to form var or pyloform coated grids and examined under a transmission electron microscope. Electron microscopical examination. Negative contrast electron microscopy has proven to be an extremely useful procedure for quick diagnosis of camel contagious ecthyma. The virus virions are ovoid particles that range in size from 230 to 360 in long and 131 to 160 in width nanometers. Their axis ratio is about 1 to 1.56 and their surface shows a regular cross pattern of filaments, pole of wool under the electron microscope. Blood chemical examination. Camel contagious eczema is associated with increased white cells and decreased RBC counts. Besides, serum biochemical tests showed significantly lower level of protein, globulin, and glucose values, and significantly increase of alanine aminotransferase ALT values. Histopathological examination. Vascular changes of the epithelial layers of skin accompanied with ulceration, neutrophil infiltration. Hyperkeratosis and acanthosis were also reported. Affected areas with ulcerated, hemorrhagic, and with frequent secondary bacterial superinfection has been reported. Immunofluorescence and immunoperoxidase are important diagnostic tests for camel contagious ecthyma disease. Polymerase chain reaction. It is currently the most popular, quick, and sensitive diagnostic test for this disease. Polymerase chain reaction process done as the following. Thermal profiles are set as initial denaturation at 94 centigrade for 5 minutes, followed by 35 cycles of denaturation at 94 centigrade for 1 minute, annealing at 55 centigrade for 1 minute, and extension at 72 centigrade for 1 minute, and final extension at 72 centigrade for 10 minutes. PCR amplified products should be checked by electrophoresis in a 1% agarose gel. Additionally, a multiplex PCR has been used to differentiate camel contagious eczema from camel pox disease as well as papillomatosis. The commonly used protocol is a gel-based PCR amplification of the envelope gene P2L gene of the parapox virus carried out using primer sequences forward as seen in the followings. This figure showed the polymerase chain reaction for identification of contagious pustular dermatitis or camel contagious eczema that seen positive results at 140 base pair. This figure showed agar gel electrophoresis of PCR product of RPO 132 gene of camel parapox virus. Lanes from 1 to 5. Samples collected from suspected cases. The positive samples were amplified at 140 base pair fragment. L. DNA marker New England Biolab. Positive control, and negative control. Sequencing of the viral genes. Phylogenetic tree based on nucleotide sequences of P2L genes from members of different species of parapox virus constructed using the Nepor joining method in Colostal X and TreeView 1.6.5 with 1000 bootstrap replicates. Horizontal distance are proportional to the genetic distances. Vertical distances are arbitrary. The numbers at each branch represents bootstrap values. 1000 replicates. 1 and 2 denotes to the groups. This is the phylogenetic tree of different parapox virus genes, where here the 1000 Colostal's view and the others we are illustrated and discussed before. Differential diagnosis of camel contagious ecthyma. The following 4 diseases have to be differentiated from Camel Contagious Ecthyma. Number 1, Camel Pox. Number 2, Camel Papillomatosis. Number 3, mange. Number 4, reaction to insect bites. Zoonotic potentiality. 2, Camel Contagious Ecthyma. None a case of Camel Contagious Eczema had been reported in people except a recent human infection in Saudi Arabia, whereas a 42-year-old man who came into contact with an infected camel developed atypical ORF lesions. Treatment. There is no specific treatment for camel contagious eczema. Nomads in some endemic areas deny the infectious nature of camel contagious eczema, and therefore the disease is either allowed to take its natural course or traditional treatments are applied. Traditional treatments include cauterization of regional lymph nodes, application of sesame oil and hot milk, and sometimes plant tar. Broad-spectrum antibiotics, either topic or systemic, given for 3 to 5 days, prevent a secondary bacterial infection and reduce the severity of the infection. Additionally, giving antipyretics, antihistaminics, and multivitamins is useful in reducing the effect of the infection. Prevention and control. There is no currently specific vaccine for camel contagious eczema. To control camel contagious eczema, sanitary measures for infectious diseases should be applied as the following. Number 1, quarantine of infected areas. Number 2, restriction of camel movements. Number 3, management of drinking water. Number 4, avoidance of skin abrasions. All the latter mentioned measures are difficult to implement in the open range production system of camels, owing to the migratory pattern of camels and difficulty to reach to them, especially during the rainy seasons. At the level of infected herds, infected camels should be separated from healthy ones and treated as described before. We keep on the track for explanation and discussing the different diseases of camels. Please subscribe the channel to reach to the newest knowledge about animal diseases.